Hi everyone. If you're watching this, you should have just watched the Amoeba Sisters video about endosymbiotic theory, and then now we're doing what will go in your notebooks. So the title can be Mitochondria, date, whatever date it is that you're actually doing this. And let's break down endosymbiotic theory first. So endo is in, sym is together, and biotic is life or living. So living together, one thing is in the other, right? The larger prokaryote engulfed the smaller prokaryote, it's inside of it. So first, let's address the word theory. As the Amoeba Sisters pointed out, the word theory in science means something different than the word theory as it's used in everyday language. So in science, a theory means an explanation of a phenomena that is supported by evidence, was tested many times, and validated by many scientists. So it's not something to be taken lightly. It's not a hypothesis. It is it's a tested hypothesis that has lots of evidence to support it. So endosymbiotic theory, what does that mean? That means a larger prokaryote engulfed a smaller prokaryote, and the smaller prokaryote remained intact and functional. So this is how we think eukaryotic organisms came to be. That's the theory, the well-founded and supported theory. The evidence we have for that is mitochondria and chloroplasts have their own DNA and ribosomes. They divide like bacteria, and they're about the same size as bacteria. We can also think about the double membrane that chloroplasts and bacteria have. So if we have a larger prokaryote and a smaller prokaryote, the larger one engulfs the smaller one, right? That's the bigger one kind of taking it in. We end up with a double membrane over here. So the membrane of the larger bacteria that was around the smaller bacteria remains, because anytime there is endocytosis, the membrane stays around what came in from outside. So we have two membranes, the one from the smaller prokaryote and this one from the larger prokaryote, all within the still larger membrane of the whole cell. So mitochondria and chloroplasts, both double membrane. This is why. I also want you to get the mitochondrial structure into your notebook as well. So let's go through that. As you can see, there's an outer membrane that I did in orange. There's also an inner membrane I traced over with purple. You can see the inner membrane has all of these folds in it. So this increases the surface area. That means more place for oxidative phosphorylation to happen. We call the space between the inner and the outer membrane the intermembrane space. So intermembrane space. I color that kind of like a brown. So this whole area here, that's all intermembrane space. The area within the inner membrane that I've colored blue here, we call the mitochondrial matrix, or sometimes people refer to it as the inner matrix. Within the inner matrix, there's DNA, just like the Amoeba Sisters said, it's free-floating, and there are ribosomes, so mitochondria can make their own protein. We also have a name for these projections. So cristae or cristae are these parts of the inner membrane that kind of stick into the mitochondria. So we have a name for those, and their role, like I said, is increasing the surface area for oxidative phosphorylation, so more can happen in the mitochondria. Thanks for watching.